But that the work that we were able to do is funded from the Environmental Natural Fund. So this that trust fund that exists through the state with the lottery dollars of environmental research. But it was brought to us because of watershed itself, and specifically the Douglas Lakes Association, Three Rivers Parks District, Capital Region. They had questions surrounding salinization to fresh water, and then we also partnered with Ramsey County, the MPCA, and then throughout the project. I said that I am part of the Science Museum of Minnesota, more specifically within our research arm at the St. Croix Watershed Research Station. And we're located on St. Croix, and we are a biologist, geochemist, paleolimnologist. I myself am a biologist. We're a team of really interdisciplinary researchers because to care about water and study water, you have to be, there's so much going on, right? Our mission is to better understand the challenges that face freshwater, specifically in Minnesota. And I will just say that I looked through our records before I came here. We have partnered to do research. Last decade, we've done 3 projects, some at historical water quality reconstruction in lakes in the watershed district, and then paleo study on. So a colleague of mine, when I said I was coming here to talk, he commented on how he's more than myself, that the Rice Marsh Lake sediment core was one of his favorite cores he's ever collected because on the lake. And there's such a clear signal of that in the sediment record. And so today, when I kind of talk about our paleo work, just keep in mind that nothing is ever forgotten, even at the bottom of the lake. Today, I'm going to talk a bit about a project that was funded 3 years ago. So we just wrapped up in August. And it was all focused on better understanding sort of the implications of freshwater salinization. I always like to start with this sort of doomsday image. It's a doomsday image for myself anyway. I'm kind of winter, but this is like bridge, and there's snow, and we're not alone. That 70% of the United States population lives in areas that are affected by snow and ice. And so when we think about that geographically, or the distribution of that, something like this. And so I will say, this is the winter of 2023. So you have to think back in your memory a little bit. We've had some anomalous winters in the past 2. But this was a year where in the Twin Cities, we saw around that 8-foot snowfall. And in response to this as a nation, so this is our contiguous distribution of snow and ice, that we would put down tons per mile of dry chemicals. And so when I say dry chemicals, I mean your like traditional road salt, so think chloride salts. And then we added another 730,000 or 7,300 gallons per mile of liquid chemicals. And so these are brining substrates that typically go on prior to snowfall events. And I'll show you. But I mentioned at the front of this that I'm a geomicrobiologist by trade. So when I see numbers like this, I think that's math. That's a big number, but I don't like have a picture of it. So I just like to convert it into something that I can visualize. And so I tend to use it as my metric for converting that. We put down 390 tons per mile. The average male African elephant weighs about 13,000 pounds. So I did a little math on the lake backyard here to see like how many elephants would you walk by if you decided to take the trail that goes along Rice Marsh Lake. So I'll let anybody guess if they want to guess. Upwards of 100, 230. That was really fast math, but it's 240. Congratulations, Sharon. In Price is Right rules, you cannot get much better than that. So if you walked along that path, you would walk past 230 elephant piles worth of salt. And so that is a lot of salt I'm not saying we shouldn't put that salt down, because we know that salt saves lives. It reduces road deaths by about 80%. This is a video example that transitioned from a brine surface than a snowfall, and you can see how that brine plays a role, sort of prepping the road for the snow. But we have gotten to this point where in North America's lakes, it is a pollutant, and it's polluting about 45% of the lakes in North America, and most of those lakes are in the Midwest and the Northeast. The study that actually found this 45% used so many lakes from Minnesota as their reference for that data set, and that's because we have organizations like yourself, groups like ours, and just broadly Minnesotans that we're so proud to be from a land of 10,000 lakes, and we will do a lot to protect them. Look at them. These are some from the Minnesota agency that have long-term. This is a lot of lakes that we have. When they looked at this data set, found that some of them were impaired for chloride. And so when you have an impairment for chloride, you are at 230 or above 230 milligrams per liter chloride. And this threshold was primarily established by toxicity. So there was some work done around this time that found what is sort of the lethal concentration or chronic toxicity concentration for some of our more sensitive organisms. You can think about bottom of the food web organisms. And some of them were sensitive at that low 230 milligrams per liter. But I always like to show this because while 230 is a good thing to set from a water quality standpoint, there are so many organisms that can withstand that. So it's good to have the bar nice and low because we put it on the list. We say, no, we're not going to get to the point where we kill all the caddisfly larvae. No, we're not going to get to the point where no mayflies can live here. And so the bar is really low for us. So when I show data and I show you that we have lakes that are above 230 milligrams per liter, I think it's good because now we know these are lakes we have to watch for. But I always like to show that the organisms that live there can handle greater concentration. And I usually give this talk to people who aren't from Minnesota. So I always like to say, if you know the geography, you'll know that the lakes that are blue tend to be of the Twin Cities. This is an urban problem. There were lots of dots on that lake. Lots of lakes have been assessed for their chloride, but the ones in the cities have the problem. And with, to a lesser extent, there's 2 lakes out in Alexandria that also have a problem. And we're also fortunate. I feel like I keep saying we're fortunate to be here, but we are because we have this chloride budget. So this was done in the early 2020s. And they found that most of our chloride that's coming into these systems is coming from road salt. So we're looking at 400,000 tons of chloride per year from road salt. And then to a lesser extent, fertilizer, wastewater treatment, and then you've got things like livestock and residential systems. And so I've told you a lot about what we know about chloride already. We know we have impaired lakes. We know where it's coming from. We know it's an urban problem. But then that's kind of where we stop. And because it's only been in the last 5 years or so that we think about this. That's where we came in at the research station and with the museum. We were really interested in understanding, so now we have this salt, what's going on? Broad approach because we're a broad group of researchers. So first, we wanted to look at it from a historical context. We have a lot of people who are interested in paleolimnology in our lab, so we were looking at the biological geochemical signatures that we could find in the sediment record during this period of salinization. Then we wanted to say, like, here and now, lakes that have salt that are really high concentrations, what's going on with nutrient dynamics? What's the interplay between salinization and eutrophication or nutrient enrichment? And then we wanted to say, okay, where are we going with this? Can we potentially predict some outcomes and better assess which lakes should we be concerned about and do some modeling work? And then because there's such a strong push statewide to really improve our salting practices, or can we explore some of the effects of, say, nutrient dynamics with these alternative deicers? And I will say that with deicers, that number 4, we had proposed to do a bunch of work looking at potassium acetate as a potential substitute for sodium chloride as a salt. And like right as we were getting the funding to do this, some work came out of the University of Minnesota that found it was actually more toxic to aquatic life than sodium chloride and created a higher biological oxygen demand. And so lakes were actually losing oxygen faster. So we had the potassium that was detrimental to the biology, and then you had this substrate availability for organisms to start respiring and using up all of the oxygen that was in the system. And so we kind of said, they did the hard work, and we'll just focus on the other three parts for this. I'll start with the historical context. So I have no idea what everyone's background here is in terms of lakes, so I'm going to pretend like you know nothing. So if you feel like I knew this, good for you, and just feel great about the day. But we're going to start really, really low, like this is a lake, and it's fall because and so from a historical context, I mentioned we go out and we take sediment cores. So if you think about sticking your straw in your milkshake as a kid, plunging the top, and then pulling it out and letting it go and watching all of that milkshake fall down, that's what we're doing. And we can just really capture a sense of what's been going on in the watershed, because every year as new sediment is falling to the bottom, it's preserving a timeline of both what's happening in the lake in that moment, but also what's happening in the watershed around it, because lakes are the lowest point on the surface. So if you change the landscape, rain comes in, it's bringing that sediment with it, and as it brings that substrate with it, it's recording that in the sediment. Typically what we do is we take different sections of that mud, and we look at a whole suite of things. The first thing that we're always interested in doing is looking at it from a time perspective. So we date it using radiometric dating. So if anybody in their home has radon mitigation system, that comes from the uranium decay series. And we actually use another decay substrate, lead-210, which comes from that same decay series. And that helps us determine it's continuously falling into our water. It's not doing any harm as lead-210, unlike when it builds up in your house as radon gas. But we can use that and we can measure that and say that this sediment at the top here was deposited in 2010. Lower is at 1950. And even lower than that we go to typically around that Euro-American settlement. So that turn of the century is what we're looking at in terms of when European Americans came to this area. From this data, we can kind of see how fast sediments were accumulating. So typically in this record, we'll see a lot of sedimentation in 1850 or right around or 1900, right when people are moving in. They're deforesting the landscape, they're changing the way soil is stored on land, and bringing it into the lake. So we tend to see really high rates of sediment accumulation then. And in all of our lakes that we saw what we call like the classic Euro-American settlement peak of sedimentation. In some lakes, we saw a secondary peak in the 1950s when we started to do a lot of industrial agriculture, and we were changing the landscape that way. And then as you move into sort of these complete periods of development, we don't really see a lot of new substrate coming into the lake. That's kind of what all of our lakes looked like. Then people who study lakes love phosphorus, and I think organisms love phosphorus. It tends to be the most limiting nutrient in a system, meaning it's the first thing that's gone. And so when it's gone, then things can no longer grow. And so if you bring phosphorus back into the system, then you tend to see regrowth. But phosphorus is also really cool as an element because it's sticky. So it's one of those elements that has a rock cycle and no real gas cycle. So unlike carbon dioxide and carbon, which has gas and nitrogen gas, phosphorus really stays in the rock cycle. So historically, the idea is that as sediment comes in and phosphorus is deposited in the lake, it should just return on a geological scale. But because of the way we do things, there's some chemistry involved that doesn't make that the case. And in fact, phosphorus can be re-released from sediments, leading to what we call internal nutrient loading. So you can shut off a wastewater treatment plant, which is delivering nutrients. But the sediment remembers there's still all that phosphorus trapped in that sediment. And during certain lake conditions, that phosphorus can back out, sort of reinvigorate the biology that's in the lake. So what we see a lot of times is we see really low levels of phosphorus when Euro-Americans come in. And we tend to see really high levels of phosphorus in the 2000s. So the turn of the 20th century, or the 21st century, we really start to see more and more phosphorus enrichment. And this is really in line with when systems became eutrophied or eutrophic. And what that phosphorus is bound to tells us a lot about the oxygen conditions in the lake. So I said phosphorus is sticky. If it sticks to aluminum, let's say, no biology really wants that aluminum. So when it's bound to that aluminum, it stays. When it's bound to things like iron, biology might want that iron, or biology is able to cleave that bond, and then they can use that phosphorus, or they can use that iron. But iron undergoes different chemistries under different oxygen scenarios. So when there's no oxygen, that iron that's bound to the phosphorus decouples, and phosphorus becomes available. So there's lots of sort of clues as to what's going on in the lake just based on the composition of phosphorus. And then the next thing we like to look at is biology itself. So we can go through and look at phytoplankton and zooplankton that are preserved in these sediments. We do this through looking at algal pigments, and they tell us what types of phototrophic or photosynthesizing organisms are present. Diatoms, which are a type of algae that have a cell wall that's made out of silica, so it's glass. So when it falls to the bottom of the sediment, you can actually see it. It doesn't decay, and nobody's using that silica. And so they make these beautiful fossils, and different diatoms prefer different in-lake conditions. So using all of these things together, we can really reconstruct what the lake is like over the course of these decades. So what we were curious about from a salt perspective is we knew that we really started to increase salt in the 1950s. Saw kind of an expanse again in the 1970s, and only in like the 2000s did we start to ask questions like, is this too much salt? And we looked at 15 different lakes across the metro area and Alexandria, and we did end up finding some salt-tolerant diatoms, so those ones that have the glass shell. And these are diatoms that thrive in very salty conditions, like borderline brackish marine diatoms. And they come online, for lack of a better word, they come online in the 1980s in two of our most impaired systems, and then in the 2000s in one system that's a little less impaired. Beyond that, they really weren't or aren't present. And so this gives us kind of an idea that when you get to the extreme, like some lakes that we were looking at, the biology will change. But in sort of the middle area where most of our lakes were, there wasn't a real sign from biology that the salt was a problem. Salt was changing the habitat. But this got us, so these are, the first ones are the phytoplankton, so the very bottom of the food web. Then we move up a level to the zooplankton, so the things that the fish are after. We were really interested in how zooplankton were responding to salt, and we did some work to try to like find zooplankton remains in the sediment, and it wasn't super successful. But so we kind of switched focus and did a little bit more molecular work. And so by that I mean like molecular biology and looking at the DNA. And so I'm going to introduce you to Daphnia. So these are two Daphnia pulex. So this would be a nice food source for a lot of fish. So if you like fish, they're eating these Daphnia. Pulex tends to be the species that exists in the east. And we have a very close relative, Pulicaria, that is in the Midwest. But biologically, they're the same for the sake of this comment. These are both female because they're a clonal population. So they're almost entirely female. They're both pregnant. But in the one on your left, the eggs are white. And on the one on your right, the eggs are green. Because those eggs came to be through two different mannerisms, parthenogenic births. So virgin birth would be the one on your left. And when the conditions are sketchy or not great for Daphnia, they will actually produce male offspring through parthenogenic reproduction. And then they will sexually reproduce with those male offspring because they make eggs that look like the ones on the right. And they have that green coating around them. And that green is a thypia. It essentially serves as like a protective shell for the organism. So what happens here is those eggs are released. They can fall to the sediment. I already told you we like to play in the mud. And so we can collect those thypia. we can actually bring them back to life. So October is just around the corner. We refer to this as resurrection ecology or zombie ecology. Really helped us evaluate how zooplankton are responding or figuring out, are they resilient to salt? And so this is work that was really led by a colleague of mine, Matt, and it was published in 2022, so just at the start of this study. So we had started doing it before this project actually began, kind of finished it at the start of this. And what we found was really kind of amazing in terms of biology. So we're going to go to Tanner's Lake, which is just off 94 in Woodbury. Like the eye of 3M beats down on it, so it's right off the highway and right near the 3M campus. And we went out again. We took a sediment core, and this time we sectioned it or took sections of it in sort of larger intervals. We found one that was, we kind of had 2016 to 2019, then a midsection of 2005 to 2010, and then 1995 to 2000. We selected all these Aphipia, these eggs that were protected or deposited during that time. We brought them back to the lab, put them in mason jars, gave them everything they could want in terms of suitable life, and we had our zombies, and we grew them, and they cloned, and we had these really strong populations of 150, 200 clones. So it took a couple weeks, and then we said, coming alive again, now we're going to expose you to salt and kill you. And this was to figure out if these ones that were deposited in that deep time interval were maybe less adapt to the salt. And that is in fact what we found, that those that were deposited in 2016 through 2019 had greater tolerance to chloride than those that were deposited sooner. And it even happened that they were greater than those that were found in the mid interval. So within a span of 5 years, roughly, this organism that we don't think about evolving at that speed was evolving to handle the salt load, an additional 100 milligrams per liter. And Matt and I are both geneticists too, so we sequenced the genomes of these. So we extracted their DNA and looked at their genomes, and what we found was that they had alterations in their osmotic membranes. So the thing that brings water into the cell, the ones that were on the top had different base pairs essentially for the genes regulating their osmotic membrane. So they were evolving to handle perhaps the load of salt that was coming in and out of the cell. This is really amazing to us, and we do still have some ongoing work that's looking at more Daphnia pulicaria across our study sites to see if any more of those Daphnia pulicaria have evolved that same sort of mutation, we'll call it, or protection against salt. When it comes to a historical context, there's really a limited salt signal. Only in our most impaired systems did we start to see salt-tolerant diatoms. But it was cool to see that in one of our salt-impaired systems, that the Daphnia were kind of undergoing this rapid evolution to increase chloride conditions, which gives hope for this idea that if chloride doesn't stop, or if we don't minimize the amount of chloride we're going in, that some biology is responding in tow. And not all Daphnia can do what the pulicaria can do, and not all zooplankton can do that, and they can't produce these eggs that come back later when conditions are good. And so just because this one can survive that doesn't mean that all of them are evolving, and that's a bigger question for more people to study zooplankton. Sort of the contemporary dynamics. Here we were really interested in kind of figuring out how salinity is impacting this idea of internal nutrient loading. So rather than thinking about nutrients that are coming from the landscape, how is salt sort of affecting what's going on internally in terms of nutrients? To get at this, I think we first just need to kind of think about what happens when salt enters the system and how it might affect the system. And we have our lake, winter now. We've got December, January, February, and we've got ice on most of the lakes here. And we've got sodium chloride building on the landscape as we're salting. And the temperature in our water is ice on the top and 0 degrees Celsius and 4 degrees Celsius on the bottom. So if you could jump in, if maybe you do, maybe you're a polar plunger, but if you did, you would be colder on the surface of the water than if you went deeper. because water is its most dense at 4 degrees C. And that's going to be important. There'll be a quiz about the density of water after today's talk. Then we transition into spring, and we start to see this huge pulse of chloride in March, April, May, when it's running in off the landscape. And this is traditionally when lakes, the temperature of the lake becomes the same. And when the temperature of the lake is the same, the lake is sort of exchanging everything. All of the water that's in the lake gets to play with each other. And as we move into our springs, or what could happen in this case, salt water is more dense than fresh water. So if you have a lot of salt water coming into your lake, that can immediately sink to the bottom, and it can start up conditions of stratification. So now we have, instead of one free to mix water column, we have a salty water bottom and a fresh water top. And then in the summertime, we get into what I call capital letter stratification, because now we have the pressure of temperature. So I said that water is its most dense at 4 degrees C, and so the bottom water of lakes tends to be very cold as the surface heats up from the sun. So if you've ever gone swimming, you dove in, but touched that cold spot, and you were like, whew, that was refreshing, or it was a bit chilly, that's thermal stratification. And most lakes in Minnesota undergo this every year. So this is not because of salt, but salt could strengthen this stratification. Then around this time of year, later and later every year for the lakes in the Twin Cities, because it's warmer and warmer later and later into the fall. But when temperatures become cooler and the top waters cool down, they become the same temperature as the bottom water, and we get this chance for the lake to mix. And if there's some salt that doesn't really change the density of the water, it's free to move back up into the water column and hopefully exit, because otherwise it doesn't leave. Things that we can't get rid of. There for the water residence time. And so however long water stays in the lake, that's how long salt is going to stay in the lake. And if it stays in the lake at too high of concentrations, we get into this thing called myrmexis. And this just essentially means that the lake is no longer mixing. So you kind of have a lake within a lake. So on top you have a lake, and on the bottom you have a lake. And both of those can have very different conditions depending on oxygen availability and nutrients, because they're no longer exchanging water. And that's just sort of a physics property. And we call it cultural myrmexis here. Some lakes around the world are just myrmetic naturally, but here we're doing it. We're adding salt, so culturally we're making them this way. I mentioned before that we were looking at 15 lakes. So we're looking at 12 of them in the Twin Cities, and then 3 of them out in Alexandria. And they range in concentration from 52 milligrams per liter to 600. And remember, 230 is impaired. So we have some that are well below impaired, some that are well above impaired. We had so many to choose from when we got here, but we decided to try to break them into sort of geographical regions based on who's responsible for salting the roads in that area, so that we could better kind of understand the salt dynamics with the lakes. And so they're grouped into 3 regions, and each region had a lake that was impaired, which is always the darker shade. So if you ever see a graph, the dark shade is a lake that we already know to be impaired, a medium shade, which is kind of close to impairment, and then a light shade, which is nowhere near impairment. We went out monthly from June of 2023 till November of 2024. And then had to skip some time in the winter, because the winter of 23 and 24 there was like no ice. So it was a super unsafe winter for us to be out collecting samples, because it was that kind of like less than a foot of ice. And I'll kind of walk through now what we found in terms of chloride. So here we're looking at our chloride data. So on our y-axis, we're looking at the concentration of chloride. And the dashed lines that you see intersecting that are those toxicity standards set by the EPA. So 230, meaning long-term exposure at 230 means something could go wrong. Acute, meaning a one-time hit at 860 is pretty detrimental to a good portion of the biology that exists. And I'll bring them in by region, and I'll start with Alexandria. And so here we can see sort of a unique pattern in terms of salt delivery. So our Alexandria lakes are the only lakes in the study that and we picked them because of this, they don't get their salt load from like road salt. They have a wastewater treatment problem. And so they know this. The ALASD, so the Alexandria Lakes Area Sanitation District, already knows that they have too much chloride coming into these systems, and they're doing their best to sort of get rid of it, but it's a water softening problem. And so you can see that in this sort of dark green one, the rate of chloride is kind of the same. But if this were, say, like one of these, you might expect in this left panel, which is the surface waters, I apologize, I didn't say that, and the right panel is the bottom water, where you see greater concentrations in the spring that then kind of go down throughout the season and then maybe pick up the next spring and come down. This is also the first time that we see a lake that has very different surface waters, so that panel on the left versus bottom waters. And this is Brownie Lake in Minneapolis, part of the Lake of the Isles chain of lakes there. And this is a lake that we already know to be stratified permanently, so it's permanently myrmictic. You can see that the concentration in the bottom waters of this lake far exceed the acute toxicity. This is a lake that is essentially two different lakes. The top of the lake is one lake, the bottom of the lake is a different lake. Two inner bodies of water that don't mix. Then from there, we had another lake that exhibited the same pattern. And this is kind of the first one where you can see the influence of winter as well. So if we look at that left panel in the dark yellow shade for Little Johanna, you can see that when we first started in June of 2023, chloride was quite high, got drawn down most of the summer as water was leaving, and then when we came back the next year in April, it was really high again. And so this is the signal of salt coming in during sort of that snow melt. And it doesn't really affect the bottom water as much because these two compartments aren't exchanging. So essentially, these lakes are three lakes within. It's a very dream within a dream kind of thing. Finally, we can see some lakes that don't have as strong of a gradient between their surface waters and bottom water. So these are lakes in the West Metro, Parker's Lake and Medicine, where we kind of see a difference in the surface waters and bottom waters. But I like to point Parker's out here, especially its bottom waters. So I mentioned at the start of this that our first winter that we went out was the year I showed you that map of data where we got 8 feet of snow. So we had a real winter in 22-23. And you can see that in how much salt is in the bottom of Parker's. But then in 23-24, we had next to no winter. And so the salt signal that came the following year was almost nothing. And so the bottom waters had a chance to recover. So there's some export of salt in that system, which is really promising. And kind of, I don't know if it's a good thing or a bad thing that we don't have winters anymore, because on one hand, we're not doing salt, but then on another hand, we don't have the same sort of physics and like meteorological conditions that are winter. And so it's like a give and a take here. So it was warmer. We used less salt, which means great, there's not a lot of salt, but that changed the conditions of the lake for another reason. And then finally, this is just the East Metro lakes. And again, the lakes that we knew to be impaired had time points where they were already impaired. When I show all of this data, a rainbow sandwich, it's super overwhelming. I'd like to kind of highlight that when we set out, we knew that we were going to find 5 lakes that were impaired. What we found was that we added 2 more. We added a lake out near Alexandria that got a huge pulse from an agricultural event in the fall. And then we had Medicine Lake that had a peak sample in the early spring of 2020. And so this was kind of hopeful for us because it meant that like the lakes that were near that threshold weren't exceeding it, but it gave us sort of an indication of which ones of these lakes is the difference between the concentration in the surface water and bottom water really steep. And I mentioned that we were interested in understanding how chloride affected nutrients because we're really interested in does chloride sort of promote like harmful algae is a good example. And we looked at everything. What we found was almost nothing. That chloride was not really related to any parameter, that the signals from climate warming and just already existing phosphorus are so much stronger when it comes to all of this. There was, however, one sort of expected finding in it was that we saw as chloride increased, so did the phosphorus that was in the bottom waters. And this comes back to this idea of how lakes stratify and how they lose oxygen. And I'll kind of walk through that part in just a second. But this gets back to this idea as lakes become more chloride rich, their sediments are going to start to release more of the sequestered phosphorus back into the system. And then as they mix, that phosphorus is available for potentially algae. It was a significant correlation, not super strong, very variable by year, as well as system. It did get us on this next step of sort of assessing how salt might impact internal loading. And again, I'm going to come back to this sort of schematic. So here we have a lake that's stratified, and this can be stratified because it has salty water in the bottom, or just because it has cold water in the bottom and warm water on the top. Doesn't matter how. In the sediments, we have phosphorus. It can be bound to lots of different things. But when the conditions of the lake sort of shift to one that now there's no oxygen in the bottom water, the phosphorus that is bound to things like iron will release and come back up into the bottom waters. And when this lake mixes again, that phosphorus comes out, it's available to biology, and you end up getting these this is what we call the late season bloom. If you've ever been walking around a lake in like late October, and you're like, wow, why is there so much like algae scum on the lake right now? It's fall, this should be a summer thing. It's because most of the lakes have turned over. They've released all of that phosphorus that was stored in their bottom water. We still have long enough days that all of those organisms that didn't have enough phosphorus to photosynthesize are now growing rapidly. And what we wanted to do with our 15 really start to assess how much phosphorus can actually leave from these sediments. So we set up some incubation experiments where we were looking at the release of phosphorus under anoxic conditions. So when there's no more oxygen, how much phosphorus comes out of the sediment versus when there is oxygen staying there. And so what this looks like in a lab environment is something like this. And so these are 6 lakes of incubations. You can see there's overlying water on the top of a sediment core. And then sediment, and that big white thing is an alum treatment. So we cored lakes that had alum treatments to try to prevent phosphorus enrichment. And we're bubbling them with different gas. So in the blue one, we were making it completely nitrogen in the water, essentially saying, okay, anything that would happen because of a lack of oxygen, say during stratification events, that's what we're going to see. And the other one being our sort of control with oxygen. That ends up looking like, from a data perspective, is something like this, where over our experimental days, we start to see more and more phosphorus accumulate in the water of those that have no oxygen as opposed to those that have oxygen. So this is what could be happening in the lakes during these stratified periods. And when we put the math to it, we can quantify how many milligrams of P these lakes are just supplying themselves. So we often try to regulate how much phosphorus enters the system, because that's good. But it's really hard to regulate the amount of phosphorus that the system is giving itself just due to the natural physics. And what we found in this particular lake is that there's a 44-fold change the minute oxygen is gone. And so as soon as oxygen leaves that system, those sediments had the ability to produce 44 times more phosphorus into the water column than they do when there's oxygen. So oxygen is not only just important for fish and keeping fisheries well stocked, it's super important for managing our nutrient concentrations. When we look at this across the sort of, ooh, that was funky. When we look at this across the salt concentrations, they're going to be left to right based on how salty they are. What we found was that it didn't really matter how salty the lake was. The lake's chloride didn't really dictate how much phosphorus could be fluxed out of the system because we have lakes that had very low salinity but had 2 milligrams per P per meter squared per day coming out of the sediments. And this again goes back to the idea of the flux not really being driven by chloride but just happening because chloride has given stratified conditions. So it's not surprising that there's no sort of correlation here, but it does give us a glimpse into what might happen as lakes transition into states of more chloride, because we can start to see, like, as chloride enters and then climate pressures warm the lake faster every spring, what's going to happen. And with the data I've showed you, it's really hard to see that, but thankfully we put some high-frequency monitoring buoys out there, and this allowed us to capture temperature going down the water column, as well as conductivity in the top water and the bottom water, and dissolved oxygen in the top and the bottom water every 30 minutes for the 2 years that we had these things out there. So this is a super data-rich thing that really gives us a glimpse at not only how does temperature dictate the biological oxygen demand and the loss of oxygen, but how does conductivity do that? And so when we started to look at all of that data and think about, like, the P release, we're going to look at some figures like this, where on the left axis or the y-axis, you've got depth. So the top of it is the air-water interface. That's the surface of the lake. The bottom of that is the sediments. And then we've got time running across the whole thing. Now I'm going to add in color, which is the temperature of the lake. So we had a sensor every 2 meters going down the lake to record the temperature. To do that, this is what we see. And so I've labeled them as stratified or mixed periods. So whenever you see kind of a horizontal line-ish, so you can, I'm going to point here, maybe I have a mouse. No, you can't. Only people in the room get to see me point. So this horizontal line indicates the period of stratification. You see a vertical bar of the same color, that's more indicative of a mixed period. So for this lake, it's our sort of classic Minnesota lake, where we have a period that's stratified in the summer, mixes in the fall, stays relatively well mixed all winter, mixes again in the spring, and then stratifies again as summer approaches. And this is great because it tells us about temperature, but just because we look at this temperature doesn't always mean that the lake is mixing. It means it's the same temperature. In the case of this lake, it does mean that the lake was mixing. So here we're looking at dissolved oxygen and specific conductivity. So specific conductivity is the measurement of conductive ions that are going through, and in freshwater systems, that's mostly chloride, like it's the predominant ion in the system. And so on your left axis, you're going to look at dissolved oxygen, and that'll be the blue line as it comes in. If the blue line ever sits in one of those gray shaded boxes, it means the conditions in that water, whether it be the surface that's on top or the bottom that's on the bottom, there's a limited amount of oxygen. So it would start these sort of reactions that maybe would release that phosphorus that we were worried about. And then the conductance on this scale is pretty standard for conductance, and so you won't really see that. And here is what this looks like for, this is Cedar Lake, again, part of the chain of lakes. And we can see in the surface water, we forgot to turn the sensors on. This is the beauty of doing actual field work. Sometimes people go out and you have to toggle a switch. So when we went out in June of 2023, they weren't on. And we went out to collect data in the fall of that year. We were like, whoa, why is there no data? And it's because I turned them on. So we started collecting in the fall. And you can see that in the bottom panel, as soon as we turned them on, there's like a big spike in oxygen. This is like right when that lake mixed. And so the bottom water, the bottom panel is really the one we care about because we're interested in kind of understanding what's happening deep in the lake. And you can see that over winter, it starts to lose oxygen just because the ice is on. So there's no exchange with oxygen with the air. And so we lose oxygen until about March or April when we see it pick back up. And that's that time period again when the lake was all the same temperature and those two compartments of water were mixing. And then again, as the lake starts to set up its stratification, by June, it was out of oxygen in the bottom water. So in Cedar Lake, from June until when we pulled the sensors at the end of October in 2024, that lake was hypoxic or anoxic at the bottom, meaning zero. So most of it was spent in a period of anoxia. And so if we looked at Cedar Lake, that was getting somewhere around the 1.5 milligrams P per meter squared per day. For 4 months, those sediments were just pumping out phosphorus. And when the lake mixed, all that got to go right back up into the water column. If we juxtapose this with a lake that is not, it might have the same temperature but is not mixing, this is what that looks like. So this is Brownie Lake, just upstream from Cedar Lake. And here we can see that the oxygen profile in those bottom waters is just a flat hypoxic line. That water never gets to see oxygen. It's never being exchanged with water that sees the atmosphere, and that's directly due to the levels of conductivity. So I'll go back to that slide. We were looking at about 600 microsiemens per centimeter in Cedar Lake. Just upstream, we're at almost 3,000. And so the level of the amount of chloride in that lake just supersedes everything else, and it has the lake no longer mixing. And so in that lake, again, we're seeing close to 2 milligrams per liter per day coming out of the sediments. But, and this is one of those, again, like, should we be happy about this? That lake's not mixing, so that phosphorus is not going anywhere. So it's trapped down there. But there are some interesting bugs that can go down and get it and bring it back up. So what we found here was that in terms of contemporary dynamics, salt doesn't matter in the short term. It's really a long-term problem. And like with years of data, you start to see the effects of salt. And really the predominant mechanism for like nutrient problems is still thermal stratification. So the climate signal is still a much stronger signal at the moment than the salt signal. And now, and if you fell asleep, I don't blame you. If you were bored, I don't blame you. But if I could bring you back for this part, this is like my favorite part of this. And it's not just because this was the part that I did in this study. The other parts are like other people. I'm not that egotistical. But this part is really good because this is like the start where we get to say, so what? Like, what does this mean for me? Like, how do I handle this data and how do I use this? And we were actually starting to model these sort of salt tipping points. So I've explained how salt might affect biology or how salt might affect nutrient status. But like, when is that going to happen? And should I be worried about it? And that's what we're going to cover next in this sort of perspective. So what I wanted to do was kind of figure out, can we quantify when there's an amount of salt in the bottom water that's going to essentially say, nope, I'm done mixing, done. And the answer is yes. And I won't go into the math, but I'll kind of walk through like how we approach it. And so this is our lake again. It's split in half because it's stratified. We know the depth roughly where that happens. And what we can do is we can look at the density of water in that surface water and the density of surface in the bottom water. And what I did is I just modeled continuously adding salt to the bottom waters until it got to the point where the density of that water prevented the lake from doing any mixing. And so this is a way that we can say this is the critical threshold for the amount of salt that would need to enter the system to prevent this mixing. That, in terms of data, we're going to look at it in salinity units. So it's not like chloride before where we were looking at like concentration of chloride. This is salinity, so it's a unitless number. But you can kind of think about it in turn with chloride. The numbers would be very similar. And so here we're looking at regions again, and they're always ordered from a lake that is not salt impaired to a lake that is. So it goes not impaired, kind of impaired, or close to impaired, and then impaired. And when we look at sort of the amount of salt that each one of these lakes can tolerate, we would have expected to see something like we see with the Minneapolis lakes, where the lake that's not impaired has a greater amount of salt than the lake that's close to impaired, has a little bit less salt that you can add, and then the one that is impaired has even less salt that you can add. It's like a little bit of a stair, but we don't really see that. And what we find is that there's kind of like a unique number for each one of these, and it's really determined on the physical characteristics of the lake. And so one thing that I have to do in this is determine the surface area of the lake, because wind is probably the most significant factor in this. So if you have a really, really big lake that has a lot of surface area, that means wind can, or a really long, we call it a fetch. So it's a really long, skinny lake that the wind can go all the way across. That lake is going to mix really well because it has a great surface area that it can go But if it's a circle basin lake, really steep sides that are in the water and is super sheltered with trees, that's going to play a role in how easily it is for that lake to mix, meaning not going to mix well, so it doesn't take much salt to really get to that tipping point. So I have to model all of this sort of with like these ideal conditions, and the ideal condition for me was a wind that was 5 meters per second, which was like the average wind for 2024. But I wanted to see, like, because we have such different meteorological conditions, and because these lakes have different basin characteristics, whether or not they have a lot of trees or are in an urban, really super urban area, so they're not getting a lot of wind or they're getting a lot of wind as a result, can we model this with respect to wind? And then can we also say, where's the lake now and how close is it to that threshold? Because these numbers that I was showing you don't mean anything if we don't know where the lake is right now. So now what we're going to look at is the lakes are again in the same order, but now on the bottom, we're looking at the ratio of the lake's current salinity to its threshold. So if the lake is at 1, it means that it's already at its critical threshold. If it's above 1, it's already above its critical threshold. So if you're in the gray box on this slide, that means you're no longer mixing. And the colors with what I just showed you. So this was that 5 meters per second. So at the average wind speed, we had 2 lakes, Little Johanna and Brownie, that are no longer mixing based on their physical characteristics and average wind speed. And this confirms what we already know to be true, which was great. It's always great when the model tells you something that you know to be true, because then you feel like you can keep going with it. And so we said, well, what happens if we have a spring that's lower winds, or one that has really high winds? How does that change? Some of these lakes, it will push them to a point where they're no longer mixing. And typically that's like a 3 meters per second wind speed, which is not very typical. We usually have windier springs than that. But what we see for the 2 lakes that we already know to be Miramichtic, Little Johanna and Brownie, it doesn't matter if it's the windiest spring on record, those lakes are not going to mix given their physical characteristics. And this now gets us to this point where we can look at lakes, we can say, how close are you to that dashed line? And should we be worried? Which ones can we target and say, hey, we have a salt problem in, let's say, we're looking at, I'll look at these ones kind of in the middle. Like we have Phelan, McCarran's, and Tanner's. Those points are all very close to that line. And if we have slightly different wind conditions, we might see seasons where they're no longer mixing. And this is great. This gives us potentially a target, but it's also a little bit misleading because I mentioned before that chloride doesn't leave. So we have historical data that shows chloride just keeps accumulating. And so what I wanted to do next was I wanted to say, given the historical data that exists because watershed districts are collecting it, sanitation districts are collecting it, or the MPCA is collecting it, how can we use that to sort of say how long before we reach that tipping point? And we couldn't do that with all of the lakes, but we could do it with a few. And this was sort of our really big thing, is before I mentioned that McCarran's and Tanner, so these ones right here, and Phelan, we're all really close to the same dashed line given their current salt and the model's predicted salt threshold. But because Tanner's is accumulating salt so much more rapidly than the others, it's actually pushed its tipping point to this year. And this is sort of corroborated by what other people who study that lake have found, that some years it doesn't mix, other years it does. And so as you transition close to this dashed line, you start to get to, even though your S critical value might be lower, if you're not accumulating at any sort of significant rate, it might take you, 30 years for McCarran's, whereas Tanner's had a higher S critical value, but it hit it this year. And so this tool, if we have like 5 to 10 years of chloride data, can be really helpful if we also have that buoy data that has really that high frequency data looking at the turnover during spring to really get us at this, like, what is the salt threshold and what should we do if we know our lake is close to it? And I don't know what we do if we know our lake is close to it, but perhaps it's thinking about, like, this is a lake maybe we want to think about buffering, or this is a lake that we want to think about removal practices. which tend to be very expensive, but still. And so with this third sort of perspective look, we were able to highlight 3 lakes really that were already listed as impaired. So technically we're already watching them, but that are really close to that physical disruption where their lakes are going to be behaving very differently. So when it was all said and done, we did a lot, and I am officially 2 minutes over everyone's time. And I'm happy to answer any questions, whether it's here now or you can always email me. I'd be remiss if I keep saying I or we, but there are so many people that have involved and touched their hands in some part of this data set because we can't all be experts in everything that I just shared. And so these are just some of those folks.
Hailey,
yeah, go Sharon.
Yes. So now it's something filings.
Yeah.
Some of our I was thinking when you're phosphorus.
Yeah, well, it's still that iron. So it's when iron switches states. So iron can be iron 2 or iron 3. And when it's iron, I always get this backwards. So it's when iron is one of them, phosphorus will bind to it. When iron is in its other form, it's aqueous. So it goes into solution. And so when it goes into solution, it can no longer hold the phosphorus that was bound to it in the mineral form. So when you're adding mineral form iron, you're providing a substrate that the phosphorus can bind to, because you've got a solid iron and the phosphorus wants to stick to it. But if the conditions change, that iron, depending on what else is in the filing. I think when they do the filings, they're like, there's an additional thing that's like the type of iron, like it has something that's not going to release it or it's going to stay solid, that it won't go back into that aqueous phase. But that's what's happening. And we can actually see signals of like those filing or those like iron sands style best management practices in our Little Johanna Lake, because we see this like huge spike in the amount of iron that's being deposited in the sediment. And the only way that iron's coming in is if upstream in the discharge like pipeline, they have these iron sands, and then all that iron sand comes into the sediments. I don't know enough about the like composition of the sand, but yeah, that is what's happening. So you're giving a substrate for free phosphorus to bind to. It's the same thing that happens in the alum treatment. You're giving aluminum to the system that the phosphorus can bind to. So everybody needs iron. I don't know if we need the amount of iron that the organisms need iron that are in there. So like iron, for example, is the limiting nutrient in the ocean. So organisms in the iron, which is why they always talk about iron feeding, where you like give the ocean iron, you fuel phytoplankton growth, you draw down CO2. So like that's the principle there. So there are organisms that probably are like, ooh, I like this iron. Like iron reducing bacteria are probably like loving it because they're like, I've got a substrate now. I think that if there was I'm sure there's studies that have looked at how much iron is too much. I have no idea what it is. And typically with the iron, it's settling out right away. So it's not going to like be in the water column. It's going to be like a sediment thing. And we have lots of iron in our rock formations, things of that nature. So it's probably not as detrimental, but I don't know. Exactly. That's a great question. Mm-hmm. Yeah. Yeah. So I forgot to ask or repeat Sharon's first question, but the second question she just asked me was, I said that we found then the few lakes that we knew were already listed as impaired, but were also at this physical tipping point. Who did we tell? And the answer is nobody yet. I just wrote this report a week ago and finished this. So like this is like very hot off the press. So our report has been published, or it's in the process of being published through the state. And then I will actually go give a talk with the folks at Three Rivers, and they're the ones that manage medicine and work with Parkers. And so they'll be very invested. And then the folks with Parkers have been working really closely with bar engineering. And so I've had conversations with them just about our data so that we can share it. And then we do the same thing with the lakes that aren't listed, right? So like it's great for the people who are concerned with managing tanners and Parkers to know that their lakes are really close to tipping, and we'll reach out to those watershed districts. One of them we already have, the other we have not yet. But we also share this data. So like the Minneapolis Parks gets all of our data on the ones that are in the chain of lakes. And then they can use that to inform future management perspectives. And like I have met with, I forget her name, to kind of like talk through how we were sampling. And now we'll have meetings now that the project is officially wrapped to kind of have report-esque data. But that's a great question, because sometimes it's hard for us too, because we're like, hey, we've got this data, the data set's published, we're close partners with people in the MPCA, but if it's not like a lake like these ones that are already listed, it doesn't make it really, they can't list it again. They can't be like this. And so watershed districts are the best people to talk to, because you're the ones that are working with people who own the land on the shore, or people who might be interested in putting forth the dollars to think about, well, how do we remove some of this chloride? And you wouldn't know that unless we tell you this data. Really close, yeah. Yeah. I think this is going to sound weird. I think it's less concerning if the lake tips. It's like if Tanner's does tip, I'd be less concerned about it than I would about those lakes that are actually like in the 20-year time frame. Because in the 20-year time frame, we could do something. Tanner's, we're at this point where like, unless we're going to spend millions of dollars to try to pull this chloride out, we're just going to have to hold steady and wait for the water residence time to flush the system, and then hopefully get back to sort of like a near neutral chloride standpoint. But for those lakes that are like in the 25 to 50 year range, if we just keep going status quo, in 25 years, we're going to have a problem. And so those are the ones that I'd like to think that like I would look at now and say, well, what can we do? And this is where the few conversations I've had with the engineers, I'm not sure we have a great solution yet, other than to be like mindful about what we're putting down, because the removal processes aren't up to speed with the input processes. And then in terms of like the health of the system as we approach it, so I kind of alluded to this, those two systems that no longer mix, they had really high concentrations of phosphorus already in their bottom waters. But the reason they're so high is that they're accumulating, but then they're not being exchanged. So they're never going back up. And so essentially that salt is acting like a cap. And so I'm not advocating for salt capping our lakes at any level, but salt is capping some of the nutrient problems that we're having. But what it's not going to do is just like solve that, because as you approach that dashed line, you're going to have years where you mix and years where you don't. And that year that you don't, you accumulate a lot, then you mix, and boom, you're going to really go. And that's going to look weird to us as researchers if we don't think about salt, because we're going to be like, whoa, where did all this pee come from? Like, did last year there wasn't this problem? Did somebody just like fertilize? Did someone just like open up a floodgate? And was there this new point source? And really it's just that, no, salt was capping it for a year, and then salt is no longer capping it for a year. And so you had this big pulse of nutrients. So I think those lakes that are close to that dashed line are going to see periods where the lake looks great. And that's because there's salt trapping it. And then the next year, wow, the lake looks like crap. And we might focus on the year where the lake looks like crap, but that's a great salt year. And so that's telling us we did good on salt, but not as good on nutrients. And so it's like a very like weird kind of back and forth there.
Well, this is
Don't get too excited, Jeff. I feel really bad because our report says like there's a lot of caveats, but yes, thank you.
it's super exciting. For me.
That's okay.
One that I'm remembering right now is I'm just curious about the of the stratification.
Mm.
You know, so I mean, like,
Mhm.
I can imagine that as it's starting to
Exactly.
kind of keep, for some of the lakes, it might be just having insight. Which one has some of those, and then you
Yeah.
think maybe things were just not, or you know, like.
Yeah, this is a great question. So Jeff is asking about, as a lake becomes saltier, we were talking about the idea of density. So the salt water is going to the bottom. And so it's quite possible that the center, or like the non-mixing volume, is quite small to begin with. It's only like a meter up from the sediment. And then the rest of the lake is still mixing or thermally stratifying at a different place. What we tended to find is that the point at which our conductivity really increases, indicating that that's where our sort of saltier water is, was at the same spot as our temperature. And so our temperature was kind of like regulating where even that, we call it a chemocline when it's induced by salt, or a halocline, and then we call it a thermocline when it's induced by temperature. We tended to see them in the same spot. But there are examples of lakes, especially as they start to try and mix, where they're not mixing completely. And this is what we saw with Tanners in particular. So when we went out in the fall in Tanners, it's like 14 meters deep. And what we found is that all summer the thermocline was sitting at like 4 meters. And that's when we would see a peak in conductivity. Not a huge spike in conductivity because it actually doesn't have a lot of salt in it, but it's a lot of salt with respect to that system. And then as the season progressed, it actually pushed the salt layer down. And so when the fall came around and started to mix, it mixed the first 4 meters. Then it mixed the first 8 meters. And then we went out in November, there was still a meter above the mud that hadn't mixed. And now we don't know if it's going to mix, because we didn't go out this year. But so we do see that. And then the same thing can happen in the spring. If you get a really big pulse, most of these systems, they kind of like, they gradient themselves as the salt comes in, because there's never a huge like load that like gets it to that 0.5 grams per kilogram or 1.5 grams per kilogram. The salt load that's coming in is relatively dilute compared to that. And so I have this demo that I do that I think actually happens in lakes, but I can't confirm it, so you just have to roll with me. It's a rainbow, but so I can make a rainbow where every color is a different density, and they just sit on top of each other. And so I think this has to happen in some of these lakes where that really dense water is just sitting at the bottom, and then the next year there's still dense water coming in, denser than the fresh water that was on the surface, but it's not as dense as that bottom one. And so it just sits on top. And so then you start to build these density gradients, and depending on the strength of those density gradients, sometimes it'll stay locked and physics will prevent them from mixing. But if they're close enough, then there's less, it takes less energy to break that barrier, and then they mix in the whole little, what was once 2 meters with different densities, mixes and becomes 1 or 2 meters again, but with slightly less dense water. I think this happens, but there's also a lot of internal mixing that goes on, things that we can't always see. We often think about just like storms being a thing that like mixes the surface water. There's a lot of like internal wave fronts that can come through and like just kind of push where that thermocline sits. But it's just a great question, because I think in the 2 lakes that we already know to be miromictic, we put our sensors in the bottom, so we were seeing this huge gradient, but I actually think we should have put one in the middle, because my guess is that like we were seeing the same salinity thing happen slightly up in the water column, and eventually then you're going to have an even bigger dense area. And so that is going to be kind of cool to see. In terms of the modeling, though, I did have to arbitrarily pick where I was defining that center of volume, and it's not arbitrary. I looked at the thermocline. So I said where the thermocline sets up, that's where I'm going to set up the volume of water that I'm going to add salt to, because that's typically how the lake works. But yeah, I could change it, and then if I do it lower, we would find that most of these lakes are never mixing, or have really much lower thresholds, which maybe we should do.
Also thinking about like the
Mhm.
question in the water.
Mhm.
But it's not mixing up, and the primary, I'm assuming, is water moving
Exactly.
out of the lake.
Yeah. Yes.
But,
Some of them like 100 years. So like some of these lakes where we like really altered them. Like I don't know the exact residence time on any of these lakes, but it's not uncommon for these systems that we've really altered, like the way water flows in and out, for them to have decadal water residence times, and many of these are on the 10 to 25-year residence times, most likely. But some of them might never really completely have enough or have a long enough chance to remove all of that chloride.
I was also curious, I know we talked earlier about aluminum. Really,
yeah. Yeah.
The lakes you that it had any alum,
mm-hmm.
Did you go lower?
Mm-hmm. Yeah, I'll show you one in this video again. So when this slide plays and you watch the video start, watch the white. There's a one in the white one in the middle. I'm going to kind of go down the core there. So they're all bubbling. Then there's like a right there. That was an alum treatment. That is Cedar Lake. And then when we look at the Cedar Lake data, we can see that Cedar does have lower phosphorus flux than like its neighboring lake of Bidet. This is a little hard to compare apples to apples with this respect because Cedar is a shallower system than Bidet. So Bidet's bottom waters tend to see more periods of anoxia and they're oxic and oxic conditions. And so lakes that tend to see changes more rapidly like that, or we have a longer period of anoxia and then some oxygen, they tend to have greater flux because they have then like a big deposit of phosphorus as soon as the oxygen conditions come back. But what we're going to do next, this is like when I started, I was the one who set up this incubator. It wasn't something that the museum was doing. So as I was putting this all together, I had questions just like you were asking because I'm very curious. There's a lot of alum still right in that core. I want to understand where the phosphorus is coming from in that core. So it still had about 10 centimeters probably of mud overlaying it. Is all of this flux coming from those 10 centimeters? Or is there still flux coming from below and this alum is spent, there's nothing else to bind to, and so it's just kind of moving up the pore water and then leaving. And so we're going out and we're doing some more experiments on some, just for a different project, but I will, and actually next week, along the North Shore and in the boundary waters. And when I get those cores back, I'm going to do what we call P fractions on them. And you would have done this if you were going to do an alum treatment, because you want to have a better understanding of your sediment chemistry. Not all lakes are good candidates for alum treatments. That's just the way the chemistry of that system works. And so what the P fractions can tell you is like how much of the phosphorus in your lake right now is actually related to this iron anoxic release, and how much of that might benefit than an alum treatment, because it'll be there, it'll be available to be grabbed, and then be sunk essentially. And so what I'll do when these cores come back, they won't have alum treatments for me to test this on, but they should give me a better sense in like I'm going to call it a natural core, but an alum treatment is still a natural core. It just has another layer in it. But in a core without an alum treatment, how much mobility does that phosphorus have? Because we tend to think about this sort of top 5 centimeters as being like the most important contributors for all of this. But hopefully we'll know. And there's 2 other ones. I think Bidet did have an alum treatment, but it was like 7 years before Cedars. And then McCarran also has an alum treatment, which we were also able to see. But in terms of like flux, McCarran and Cedar are lower, but they're really within the ballpark of other sort of fluxes that we see in different lakes across the state.
I was one of
Yeah, go for it.
imagine scenarios.
Yeah.
To be able to. Then I think all the mixing
Yeah, I think
kind
we're,
of non-biological. And,
yeah. Just makes more system more turbid, yeah, and so then you end up with like different problems.
around the lake just to mix it up.
I like that. It's fun then too.
Yeah. So anyway, I mean, so, but more seriously, I mean, like, some sort of mix.
Yeah. Part of the, I'm hopeful that like we'll get enough preliminary data with these cores that we're taking, plus these cores, but I'd like to ask for more LCCMR funding to kind of think a little bit about what you're saying. We often refer to this as being like this anoxia problem because, oh, iron is no longer binding to phosphorus, so we're losing this. And so we always like point the finger at the iron. There's a huge fraction of phosphorus that's bound to organic matter that when the system becomes anoxic, you open it up to a whole different group of bacteria that are excited to get that carbon or that phosphorus for that matter and cleave those bonds. So I'm really curious about, because of the geomicrobiologist in me, how important are bacteria, and do we see a relationship between the types of bacteria, the predominant bacteria that are in those top 5 centimeters or at a millimeter resolution in the mud that actually can dictate how much pee might be released? And do we start to see that, like, if we have a group of these 25 organisms, similar to like if you think about what they do in the human body, right? Where they can be like, oh, your gut microbiome is these organisms, which is why you have IBS. It's kind of like I want to do that, but in a lake and thinking about the mud and the flux of phosphorus. And I think that that could be really helpful because when we do these site specs, we're thinking like, oh, am I a good candidate for alum treatment? Yes, you might be, but also you have a consortia of bacteria right here that are going to prevent this iron from releasing because they actually, or they are going to promote it because they want the iron or something to that effect. Or you have a lot of phosphorus bound to certain carbon substrates and you have a lot of methanogens. They're going to want that substrate and subsequently release that peat. So I think there's a really cool opportunity to think about bioremediation processes when it comes to sort of not only maintaining, well, probably not maintaining anoxia because these would be anaerobic organisms, but thinking about how we could promote the right type of anaerobic organism. That sounds cruel to bacteria, but maybe some of them are better than others at protecting the nutrient release. So that's a good thought.
Thank you.
Yeah, my pleasure.
Thank you much for coming tonight.